Scott,
Good
how
afternoon.
are you, fine sir? Thanks for joining us on this Giving Tuesday, taking a little time out of your busy schedule, your busy life. I totally appreciate all the work you do and the time you're giving us today. So welcome.
Thank you. My pleasure to be here. Good to see you again.
Yeah, you as well. I mean, I always say it every time you're on this stuff. It's like people know Scott Harper. You are leading off. And in my world of baseball, that's huge this hour. So tell us a little bit about what your team is working on right now in the lab, where you're at, what's going on. People want to know. I know they want to know.
So we're primarily a gene therapy lab. And what that means is we're trying to develop treatments for FSHD that, if they worked the way we want them to, would be a one and done. So it'd be a single treatment that would offer we're reluctant to use the word cure, but
Right.
in absence of a better word. So that's what we're trying to do. There are challenges with that. We have to get it right the first time. And because we don't get a second chance. And so, that's primarily what we're working to do. We're trying to develop a gene therapy approach that can target this gene DUX4, which is really the underlying cause of FSHD, and we want to try to eliminate that.
So what is your focus then? Excuse me. What's your focus to turn that research into treatments? You're saying gene therapy.
Yeah.
We do know about DUX4. Most of us have heard at least the basic science of it.
Sure.
But we've also heard other scientists come on with their angles, with their way or their, I guess, attack method to how to find that treatment. So where are you lining that up in your lab?
Yeah, I think all of us are focused on trying to eliminate this gene DUX4 in some way or another. And, our approach is to use, like I said, gene therapy as our delivery route.
Okay.
Our approach really is we're developing, and we have developed something called a microRNA. And it's a small piece of DNA that we can insert into a virus. And we grow this virus in the lab. We've shown this many times in mice. Now we've actually shown it in human cells donated by people with FSHD that express this DUX4 gene. We deliver these to these different models, the cell models and the mouse models, inside a virus. That virus can deliver their natural viruses called adeno-associated viruses, which we abbreviate as AAV vectors, or AAV. And these AAV viruses naturally have developed over millions of years ways to enter muscle cells. And when they enter muscle cells, they are able to deliver this DNA sequence we've inserted inside them. And then we use essentially the cell's own machinery to produce a therapeutic microRNA, which operates to reduce DUX4 levels and then offers a benefit. And when we've delivered this system to, like I said, human cells or mice, we can pretty much wipe out DUX4. The trick is, getting it, actually be able to deliver enough quantity of these viruses to be able to hit, essentially every muscle cell in the body. And in a mouse, you can do it because mice are small. It's a bit more challenging as we move up the evolutionary tree, and now we're talking about doing this with humans. So right now we're focused on the doses that are required to be able to hit substantial amounts of muscle in a human being are very large. And so what we're trying to do is make sure that our approach, because we only get one shot at it, is safe to use in humans. And that's sort of where we are. That's been our bottleneck.
So if you attack the DUX4, you said, wipe out DUX4, works in mice, and that's understandable. Good analogy of, the different size of creature here. So need more probably kick if it's a human. So what's that safe, I guess, viral load? Is that right? Is that what you're trying to almost find? Like what will work, but yet be safe at the same time?
Exactly. Yeah. So, this is not gene therapy for muscle disease is not unprecedented. There aren't approved products yet on the market, but there are a number of clinical trials that maybe some of the listeners have or
Mhm.
viewers have heard about. Some of the most well-publicized ones are for Duchenne muscular dystrophy, delivering a microdystrophin gene to try to treat Duchenne muscular dystrophy. So, there are 3 companies that are leading that effort. They all have their own versions of this gene therapy approach, but they're using similar viruses that we're using. The strategy is a little bit different, but the aim is the same, which is to, package a therapeutic DNA sequence inside these viruses and then deliver them to people at high quantities so that, these viruses can hit enough muscle cells to offer some benefit and deliver that therapeutic payload. So, the DMD trials are establishing the precedent that this is doable. The companies are Sarepta, Solid, and Pfizer all have programs. And, we're waiting on that data to be released. But, from what I'm seeing, what I'm hearing, there's some promising results. And what we're trying to do is use the same sort of delivery system to be able to deliver our gene therapy to FSHD patients. The doses required are substantial. I mean, they're high. And like I said, we want to make sure they're safe.
Is this something then, and now this might be tough for you to answer because you're not there yet, but in theory, is this something then that someone would have to go get repetitive treatment? Or is this you get a one-time treatment and it knocks it out?
One-time deal. That's the great promise of gene therapy. If it, theoretically, if it works the way you want it to, and I say theoretically because there are so far only a couple of approved gene therapy products on the market right now. One's for a blindness disorder. Another is for a spinal muscular atrophy, which was actually developed in Nationwide Children's Hospital here. Not by my lab, but Brian Gaspar and Dr. Jerry Mendel developed that. So that was shepherded forward by a company called Avexis and Novartis. So anyway, there is precedence for gene therapies being on the market, but these are so new still that we don't know, how long they last, because the patients that have been dosed with those first versions of gene therapy have only had it for a few years now. But theoretically, the way this works, these viruses are stable in muscle and nerve, especially. So, you could have a one-time treatment and have lifelong benefit. That's the hope. In practicality, it might be necessary to redose later on. But the way we're thinking about this is a one-time treatment.
And when you get the treatment, is it stopping where the disease, is right at that moment? And is it also helping reproduce muscle, or rebuild it, or no?
No, well, it's an FSHD is sort of an interesting case because this DUX4 gene, maybe some of your other speakers have already talked about this a little bit. It's not on in every muscle at every given time, right? So it's a really puzzling thing, actually. You would think that FSHD can be very devastating, as I'm sure you know, and so you would think that this gene DUX4 would be on everywhere.
Right.
But it's actually not. So there seems to be this accumulation of DUX4 over time, and DUX4 causes damage to muscle. So what that means is that a person who has FSHD, if you could take a muscle biopsy from them and look at that muscle biopsy under a microscope, you might find DUX4 is present in only a few of those muscle cells at any given time, at that time of the muscle biopsy. So what this means is that if we can give a treatment to these cells that aren't expressing DUX4 at that time, then our therapy is stable and it's present there in those muscle cells. It's sort of laying in wait for DUX4 to turn on, and then it can attack it virtually immediately. So in a lot of cases, I think, we can think of this as a prevention. And what that means is that I think we would we'll be able to stop the disease from occurring in those muscle cells where DUX4 already doesn't exist and where it might exist in the
Okay.
future. Whether or not that therapy can restore function, it improve, it's possible. And the reason I say that it's possible is because we know that human muscle has the capacity to regenerate itself a certain number of times. And so if you've had a muscle that's been damaged, a muscle cell that's been damaged by DUX4, and maybe that muscle cell has died,
Okay.
we have muscle stem cells that can try to repair that muscle. Okay, so now let's think about this situation where we've delivered our gene therapy to a human. We've got the gene therapy being expressed in lots of muscle cells, and it's able to prevent damage in the cells that don't have DUX4 and then turn it on later. Okay. When muscle regenerates, so and then we have the muscles that have been damaged. Those muscles that have been damaged can regenerate themselves. And when they regenerate and incorporate back into that muscle group, they might benefit from the therapy that's already there. And so I guess what I'm trying to say here is that, and maybe I'm not doing it as well as I should, but what I'm trying to say here is that by taking advantage of this preventative nature of the way we're doing this gene therapy, and also take advantage of the capacity of human muscle to regenerate itself and reincorporate a muscle cell into a whole group of muscle, it's possible that our therapy can benefit regenerated muscle as well.
Wow.
Meaning that you could have some restoration of function instead of just an arrest. But that remains to be seen as something we have to test.
Right.
And it's harder. It's a harder question. Yeah, exactly. And it's a harder question to answer in a mouse, because mice can, mice only live 2 years, but they can regenerate muscle essentially their entire lives. So, humans have only a restricted capacity to do it. They can regenerate a few times and then they can't anymore. So it's really a question, I think, that's going to have to be answered once we actually do the clinical trial.
In your work at your lab, what's kind of been the biggest challenge over this past year?
Well, COVID was challenging, but I think our institution did a really great job of managing that, and we were down for a couple of months, but then we were able to get back and work in a restricted fashion. So I think this was happening a lot across the country, where we had shifts, we had a certain number of people that could work at the lab at different times. So my lab was very, my lab personnel were great about being able to adjust to that and be able to be flexible. So we were open 24 hours in my lab.
Wow.
So that was challenging. The other challenge we're faced from a COVID standpoint is, I think this is happening, across the board in all industries. The supply chain problems are an issue. I mean, we're down, just even basic supplies are back ordered for months, and so we've had to adjust on how the kind of experiments we can do, just because we can't get supplies. So that's been a challenge. And then, but we're still cranking along here. And the other thing I would say is that it takes money to do. We're at a stage now in our program, and we were last year when I talked to you about this, where the things we're doing now become really, really expensive. And so we've partnered with a local biotech company here, a startup called Armatus Bio, ARMATUS, and they're helping support our lab and helping us raise money to actually develop, bring these things to the clinic. But it just takes time to get those things up and running. And so the funding is coming, but it's slower than I want it to be.
You find then that days like this, Giving Tuesday, other type of fundraising events is crucial to kind of keeping that flow happening?
Absolutely.
Yeah.
FSHD Society has been a supporter of my career in my lab from the very beginning, and I can't thank them enough. And really, this therapy that we're trying to translate now, that we're at this point where we're doing extensive safety studies, that initiated from an idea we had, about 14 years ago, And the very first grant we had funded in my lab when I started my lab about 14 years ago was from just a small $10,000 grant from the FSHD Society.
Oh, wow. Yeah.
And you know, we use that as a building block to get NIH money, and now we're getting private money to try to get this. Like I said, we're in the more expensive stages of this therapeutic development, but really this all started with really a comparatively small grant. I mean, $10,000 is a lot of money to most people, but for a research lab, that's a really pretty small grant, and it really kick-started us on this strategy.
Most patients out there is listening, understanding the science of it. You're right. We've heard from other experts, other researchers and whatnot. And it always seems to always have that underlying, how long do they have to wait?
Yeah.
Or for me, it's almost a better question of, do you feel at least we're still on pace? Mark Stone's always, said 2025. We'd like to have treatments by 2025. I'm okay with, no one giving me an absolute, because I know that that's just not how this works. But do you feel that we're at least headed in the right direction, on pace with what's going on right now?
Absolutely, I do. Yeah. And it's been, the last, so, I'm confident in what we're doing, just my lab personally. Our approach works. It's like I said, it's for us, it's a matter of making sure it's safe before we go in. But, just attending and actually helping to host the last International Research Congress and seeing all of the new companies that are getting involved in FSHD, the new approaches to inhibit DUX4, it's all very exciting. And I think having that critical mass of minds and dollars and different groups working toward the same goal, it's exciting. And I've been working in this field for about 14 years, and when I first started attending these meetings, there was no industry there. It was all academic labs. So to see how much it's changed, the attendees and the presenters at these meetings is very exciting for the field.
That's amazing. That's great to hear. Yeah, because you've been in this for a long time. Some might argue from the beginning, so to speak, and to have your perspective of where it was to where it is now, that is very promising to hear. And it's good to know we're on pace. That's good. That's good. So when we turn the calendar over to 2022, what are some of the goals that you have set maybe in your lab that you're able to share with us that you're hoping to hit certain markers throughout the year?
Yeah. There was one of the viruses that we're using right now, it's called AAV9, and it's been tested in the clinic. Actually, the spinal muscular atrophy gene therapy product that's approved, FDA approved, is an AAV9 based product. But we do know that, there have been other gene therapy trials for muscle where people have used high doses of AAV9 and have had some adverse events. So that's why we're concerned about the levels of virus load that we need to deliver to achieve the effect we want. And AAV9's it works really well, and like I said, in mice, and there's an approved product in humans that uses it. But the levels that are required for widespread muscle delivery in humans is very high. So there was a paper that came out recently. It got a lot of press. It was featured in the New York Times, where a gentleman at the Broad Institute, MIT, who actually I think has a family member with FSHD.
Yeah, he was just on the last hour.
Okay, good. Sharif, yeah. So Sharif has this paper that was just published, and he started a company where he's developed some new AAV viruses that, the paper shows work 10 to 100 times better for muscle delivery than the AAV9 system that we are currently using.
Wow,
that's game changing. I mean, so we want to in 2022, and we're working on these now. We're trying to put our therapeutic payload into his new systems and determine if we can use that 100-fold less dose. And if that works, I think we're ready to go.
Wow. I think I just got goosebumps right there, Scott. That's good stuff, man. You just brought the two the two worlds together of what he's doing and what you're working on, and merging those two and seeing how that is actually, man, that just feels great. That's just great news. That's great news. That's good. That is good, promising news that you're sharing with us. I appreciate that for sure. Wow.
Yeah, we're excited. We're excited to try it. And, like I said, that paper is fantastic. And I think it could be transformative, not just for FSHD, for all muscle gene
Yeah.
therapy programs.
I'll tell you, sir, I don't want to keep you much longer. I know you got to get back to work. I know probably some of the people even listening, watching are going, all right, that's great. Let's get them back in there. Don't have them waste any more time. Thank you for being here on Giving Tuesday, spending more time with us, letting us know how your lab's doing, what's going on in your world. We appreciate it. As always, thank you very much.
My pleasure. Good to see you again.
You too, Scott. Thank you.
All right. Bye.
That is amazing news, folks. Yeah, because we just had Sharif on earlier and talking about what could be impactful on this Giving Tuesday. So you have Sharif in the first segment talking about his delivery system, and then Scott saying how his treatment could be on his delivery system, basically, to help merging of the world. So you can see, the research isn't in silos with one doesn't know what the other is doing. There's definitely connectivity. It's amazing. So I hope you all enjoyed that interview as much as I did, as well as the others we've had throughout the day.